Hey, I'm Jeremy Pruitt, and I'm talking about the article Targeting Proinsulin to Local Immune Cells Using an Intradermal Microneedle Delivery System, a Potential Antigen-Specific Immunotherapy for Type 1 Diabetes. So this article, I have the authors listed there. They are from the School of Pharmacy and Pharmaceutical Sciences of Cardiff University in the United Kingdom, the School of Medicine from Cardiff University, the United Kingdom, and from Biome SA from Brazil. The research is part of the EEASI European Research Network, supported by the European Commission under the Health Cooperation Work Program of the 7th Framework Program, and is also supported by the Engineering and Physical Sciences Research Council and the Juvenile Diabetes Research Foundation. This article was accepted on the 17th of February of 2020 into the Journal of Controlled Release. It was published in volume 322. This journal has a site score of 14.2 and an impact factor of 9.776. Type 1 diabetes is an autoimmune disease involving the pancreatic B cells, the insulin-producing cells of the pancreas. And it's caused when the T cells destroy those pancreatic beta cells, causing hyperglycemia. The skin is an effective target for antigen-specific immunotherapy, which induces regulatory responses via tailored dosage and delivery route. Microneedles are a high molecular weight delivery system. You can use these with compounds such as insulin, and delivers the compound through the skin in a minimally invasive and painless manner. Coated microneedles are a subcategory of microneedles where the formulation is coated on the microneedles, and after puncture, the compound simply dissolves into the tissue. Proinsulin is the precursor to insulin, and is an autoantigen implicated in type 1 diabetes development, but it poses a potential therapeutic character. The proposed hypothesis for this research is that immature steady-state dendritic cells and/or the epidermal Langerhans cells that display tolerogenic functions will be targeted by the proinsulin-coated microneedles. Tolerogenic means that it produces an immunologic tolerance. The microneedles were composed of 10 rows of needles, roughly 500 micrometers long and approximately 200 micrometers wide at the base. 3 rows were used to produce the 30 punctures as shown here in this picture with the mouse on the mouse's neck. The coating formulation had a concentration of approximately 82.9 milligrams of proinsulin per milliliter of the solvent. The solvent was a 0.2 molar acetic acid and 0.5% polysorbate 80 solution, and approximately 31.5 micrograms of proinsulin was indicated used. The proinsulin-coated microneedles were applied for 1, 2.5, and 10 minutes to the necks of the anesthetized mice. Mouse spleens were harvested and processed into a suspension, and the G9-CD8 plus T cells were isolated. They're incubated in a 2 micromolar CFDA SE, which converts intracellularly into a fluorescent CFDA, allowing the cells to be detectable by flow cytometry. The G9-CD8 plus T cells recognize the B15 through 23 peptide of proinsulin, and they also proliferate in response when they're presented by DCs. or dendritic cells. For the most part, 26 to 30 out of the 30 microneedles would pierce the skin of the 6 mice used. The highest average delivery efficiency was 86.1% at 2.5 minutes. As you can see on the image, on the top image on the right, the estimated PI dose delivered to the skin. It was also the most repeatable. SEM images confirmed proinsulin delivery from MN, and it was easily detected. The proliferation of the G9CD8 plus T cells was easily detected in the MN treated groups, as you can see in the lower figure. Overall, this study explores coated microneedles as a potential delivery system for proinsulin, which is overall an advantageous ASI candidate. Microneedles are beneficial due to the enhanced stability of using a dry drug form. Local immune systems are targeting was confirmed by the results and seems to support microneedling as the cause. The alternate hypothesis of the proliferation and the effect that the drug had on the mice is that it's possible that the proinsulin was not taken directly into the antigen presenting cells, but instead diffused into the lymphatic vessels, which would drain into the axillary lymph nodes. For the CRAP test, it is a current article published in 2020. It's very relevant as it's a research for a type 1 diabetes treatment. The authority it's a reputable journal as shown by the site score, and the institutions are reputable as well. It's an accurate article with plenty of functional resources and links, and the purpose again is to inform of a potential type 1 diabetes treatment. And this is the source for the article.